Alrighty, let's get started. So, who would have thought we'd make it week 3, hey? I'm tempted to make a Fight Club joke because there's like a good chunk more people here this week than the last couple of weeks. So hopefully, if there are any questions that pop up, please just sort of jump in and ask me to clarify. If not, there are recordings of the lectures that I will share or Archie will share. I think we've got a couple of them truncated, but they haven't been shared around yet, right? Globally? Okay. We'll wait till the end and we'll put something on a cloud somewhere and you can all look at them. All right, so the first week we talked about kind of gross macroanatomy, where the lumps and bumps on the brain, what all the little sort of bits and what are their names. And then last week we started talking about microcircuitry, sort of went the exact other direction. We zoomed down to the level of cells and tried to ask questions about whether or not different parts of the brain, particularly we focus on the cortex and the thalamus, had particular wiring principles. Before I get into my full recap of last week, James came up after the talk again and told me that the cortex is not the size of a rug. And so I had a sort of pretty standard reaction to that. It actually is the size of a bath mat. So thanks again, James, for making me look like an idiot in front of everyone. So recap. So what we talked about last week was this idea that you could separate out and sort of parcellate the brain, in this case the cortex, into these different zones that have different microarchitecture. So the idea is that back in the day, Corbini and Brodmann looked under the microscope at sections across the cerebral cortex, this kind of lumpy exterior of the brain, and he looked down and said, oh, I can draw these kind of artificial but defensible lines between areas of the cortex in which the cytoarchitecture changes. Maybe you've got more of one cell type over here, and then you move over and you've got a different cell type. And so if you see things like Brodmann areas, that's kind of what they're referring to. We zoomed into a extremely simplified view of the cerebral cortex. The idea was that we have these 3 kind of main layers in the cerebral cortex. We have a granular layer, which is described as such because it has a granular appearance on particular types of staining. That's an area where there's this type of stellate cell, which I'm not showing you here, which is a form of a pyramidal cell, just like these 3 that I'm showing you here, without really big, long branches on it. And it receives the main thalamic input from the core thalamus. And then once that input's come in, it gets directed to these different channels. There's certain rules. The cortex really, when you look at it, looks like a big thicket and everything's connected to everything. But there are rules in terms of which direction the flow of activity can go. And so we talked about this idea that a thalamic input might come in. It's going to hit those stellate cells I talked about, but also the apical dendrites of these CT-type cells, which are we call the corticothalamic type. From there, they'll propagate down to the thalamus, and then more often than not, we'll get a reciprocal connection back that will then augment that activity and maybe cause a little resonance. They'll also project forward to the supragranular layers, which are the areas that are above the granular layer and tend to be where all of the connections between cortical layers occur. This here is what we call an intratelocephalic type or an IT type neuron. One point that I didn't stress last week that I sort of thought about when I went away that I wanted to make a point of now is that when you see the names of these different cortical types, they're actually named according to where the cell body lies, rather than where their dendrites lie. And there's actually it seems like a kind of finicky point, but it's important to note because, for example, the guy on the right, this what's called a pyramidal tract type, a PT type layer 5 cell. This is like the workhorse of the cerebral cortex. Has its cell body sitting in layer 5, and a lot of basal dendrites in layer 5. But a lot of the functionality of the cell is defined by its apical dendrites, which sit up in the supragranular layers. And so this will get classified as an infragranular cell, but it has a supragranular functional mode. And it's really important to keep that in mind. We might touch on that at the end of the talk if we have time. All right, so we've got this IT type with its apical dendrites sitting up in the feedback layers. It then projects forward in the hierarchy to more agranular cortices and does a little bit of this feed forward driving into the granular layer, and also goes down and nudges this pyramidal cell. The layer 5 pyramidal cell will both project back down the hierarchy towards more granular layers, And it does so in a relatively diffuse fashion. I didn't talk about this much last week, but one of the sort of interesting mysteries about the organization of the cerebral cortex is that the connections that are feeding up the hierarchy away from the sensory periphery towards the association cortex tend to be quite specific, but the feedback signals that come back are relatively diffuse and are spread across what we would call functional zones. That's a really interesting mystery to think about a little. The other thing that this big cell does, this layer 5 cell, is it sends off what we call an efference copy or a corollary discharge. It's a signal that goes down into the brainstem, into the thalamus, the superior colliculus, the basal ganglia, and down into the spinal cord. And in some ways of thinking about this is sort of controlling the movement of muscles, this sort of particular twitch as I move my fingers around to augment a point. But it's also warning the brain that something's going on. I've made a movement, I've made an action in the world. Quick, give me some feedback whether that was a good thing to do or not. Tell me whether or not in this context, I've done something like this before. And so a lot of what I'll talk about today is where are those efference copies going and what kind of information does the cortex get back to help shape its evolving activity. We talked about the fascinating fact that these low dimensional gradients that we see when we do brain imaging experiments that differ from the sensory cortices through to the more agranular limbic cortices are also reflected in differences in the cytoarchitecture. So in these areas that are stained as yellow or orange, they have these really thick granular layers, teeny tiny little layer 2, 3 cells, and relatively few layer 5 cells. And if you move out to the other end of the hierarchy now into these sort of dark purple areas, you get these really big layer 3 cells that have tons and tons of connections. Someone's actually shown, Martin van der Hoevel's group showed that if you look at the estimate of this microarchitecture versus a graph representation of the resting brain, you can actually see a really strong correspondence between the thickness of this layer, the cell count, and what we call the degree of a network that we would calculate from brain imaging data, which is really interesting. We talked a little bit about interneurons. We didn't really go into a lot of detail, but I just wanted to flag this again because I think this is a really important area that is receiving a lot of work at the moment. The idea is that these pyramidal cells that I've shown you here, represented as E, we've sort of collapsed them all down into one population, are under the control and control of control and control and control of control of these interneurons, these inhibitory interneurons. And so one mistake when you're thinking about the brain is to assume it's a set of these little feed-forward knock-on chains. Really, what's happening in a lot of the circuits that we see is that we've got a system with excitation that's exciting an inhibitory neuron that's also inhibiting it. And then when you layer on inhibition of that circuit, you get to these really interesting kind of high-dimensional dynamic regimes that can arise. And then you can imagine that coming in and tweaking one of these different cells, maybe you've got a direct excitatory connection to here from another area, can change the processing mode of that circuit. This is a really important concept when you're trying to think about how these systems are organized. And on the right was just showing you that one of the canonical examples here is that these guys here, these fast spiking parvalbumin cells, which are this FS here, and the pyramidal cells somehow work together in a circuit to give rise to things like the gamma synchrony we see in a lot of attentional and cognitive tasks with EEG measurements. And really, like overall, this sort of framework is consistent with an idea that Marcel Messelem pitched about 20 years ago, which is that there's this interesting gradient between the contact that we have with the world. This might be visual information, sensory information, or auditory information, and the kind of goal state that we're in. On the right, I had Maslow's hierarchy, the idea that if it's really hot outside, then that's the only thing you can think about, or if you're really hungry, that drives you. But once all of those baseline functions are kind of equilibrated, you can then start to focus on more and more abstract things, like doing a job that you really love, or waving your hands around about neuroanatomy. And then the idea is that what is in the middle of those two, at these two ends of this low-dimensional gradient, are the circuits that you need to help you work out how to achieve this particular goal, given these set of opportunities in the world. And I think this is a really nice low-dimensional kind of model for you to think about how a brain is generally organized. If people are interested, I can pass on more references to this stuff. We also covered the fact that underneath the cortex, right, this thing that people take as the brain, there's this other really rich, detailed subcortical structures that share important connections with the cortex and likely constrain and augment its different functional modes. So the one that we spent a bit of time last week talking about was the thalamus. Thalamus is sitting on top of the brainstem, like a little ball of ice cream on an ice cream cone. and maintains connections to the entire in the green, we would call that a core thalamic cell. And what it's doing is it's receiving usually a glutamatergic input, often from the sensory periphery, and pushing it up into the granular layers of the cortex. I showed you those before. There's that layer on the left-hand side. But there's this whole other circuit type, and you have to take this all with a grain of salt because it's not as clear as just one and two, but it's a sort of two ends of the spectrum, where these cells now will project up in a much more diffuse manner and sort of sprinkle their activity into the supragranular layers of the cortex. So they're much less consistent with the idea of passing a specific message and much more of a kind of general broadcast, a general kind of alerting signal, a kind of raising of the temperature of the cortex. So that's what we were up to. And a really key question then becomes, all right, if we've got the thalamus here and we think it's constraining the cortex, which we think has these really interesting gradients in it, what is coming into the thalamus, right? What activity patterns are being fed into these different circuits, the core circuits and the matrix circuits, that can then augment or shape the processing that you see in the cortex? And so I'm going to just split these into two separate kind of qualitative groups. I'm going to speak a lot about the second group, but just want to sort of briefly mention the first. So a lot of what we know about the thalamus comes from work in the visual cortex, where we will take some visual input via the lateral geniculate nucleus, which is this tiny little sensory nucleus in the thalamus, which then we can put an animal in different contexts. Can it see this image or not? We knock this out. If we put GABA in, what changes? And we know that this same principle of a visual input coming into the LGN goes to V1. So this is like a sort of basic visual circuit, is replicated for the other sensory domains as well. So the sensory input will come in via the dorsal column tract. It'll go into the lateral dorsal nucleus and then project up into S1. And the same with auditory input through the cochlea will come in through the MGN, which is the medial geniculate nucleus, and into the auditory cortex. Importantly, these all have a topography to them that mimics the inputs that they receive. So there's like a tonotopy here and a retinotopy there and a somatotopy there. The idea is that regions that are right next to each other share some relationship to the way that the information came in. So if I was to, strike my index finger, that'll wiggle a part of my LGN, my lateral dorsal nucleus and then S1. And if I do my middle finger, that'll be next door to that in that nucleus. All right, so this is the sort of the basic structure of the way the sensory receptors work. There is more subtlety to it, but just for the purpose of this talk, let's just sort of put them in that category. So then the other category is actually really fascinating because there's these two massive systems, massive workhorses in your subcortex that often get either looked at on their own or ignored by the majority of people that do brain imaging studies or think about neuroscience, but actually have really, really well-constrained, well-conserved over evolutionary time inputs to the thalamus, and that thus have different impacts over how the cortex can actually function. So the two are the basal ganglia and the cerebellum, and I'm going to talk about the basal ganglia first. So this is a little sort of zoom in of the brain. They've made the cortex transparent, and you can see these sort of funny little purple and blue structures in the middle of the brain. They're actually bilateral, so they're actually, there's a few different nuclei, but together they're called the basal ganglia. The idea is that they're basal to the cortex, and there's a ganglia because it's a sort of group of these different nuclei. Some of them are nestled in on the ventricles. You can see this little horn-shaped guy here. So one of the nuclei, the caudate nucleus, is sitting nestled into the ventricles, and then there's another area on the outside called the putamen. These are both part of what we would call the striatum. And then deep to those, you can't see them in this picture here, but sort of deep to them in a little stack of ever smaller Russian dolls, is this area called the pallidum or the globus pallidus. So typically when we think about the basal ganglia, so this is now showing you the same kind of thing. Imagine that I've made the whole thing clear now and I've just got a little two-dimensional representation. Now what I've got is my basal ganglia circuit. And if you go look in any neuroscience textbook or any medical textbook, they love to sort of flaunt this little circuit as if it somehow like makes everything clear. Now, what I remember from med school was just being like horribly confused by all the different arrows and circles that were going in different directions. And so I don't want you to, in any way, shape or form, feel like you have to memorize these kinds of circuits. Instead, I want to kind of give you an intuition about what they're trying to achieve. And so the main idea with the basal ganglia is that they've got this constant inhibitory control over both the brainstem structures that are controlling how I'm moving around, but also the thalamus, which is then propagating back up to the cortex. As you can almost think about this as a little handbrake that's on. When I wake up in the morning, neurochemicals flood my brain, and the globus pallidus starts to oscillate, and it's spiking away really, really fast. And what it's basically doing is telling everyone around it to shut up. It's like that really annoying friend. I heard it, what was the description I heard? It's sort of like this this buzzkill, right? You're like, hey, what do you think, basal ganglia? Shh. It's constantly telling you to be quiet. And then what you can do is every now and again, you can ask this other inhibitory nucleus to come and tell the shusher to shush. So that means that all of a sudden, a pattern can flow through. You can wiggle your toe, or you can allow the processing to occur in the cortex, and all is good. There's a separate pathway, so we call that the direct pathway. There's a separate pathway which runs through another globus pallidus structure. Even though they have the same initials, they're very different structures. We would call this the indirect pathway. I haven't shown the direct link here, but the idea is that the striatum can inhibit it, but it inhibits the globus pallidus, and so it has a control in a different way. Instead of facilitating function, it actually impairs it. And the idea is that you've got this sort of toggle between these two. So people often describe this as a go pathway for the direct pathway, or a facilitatory pathway, and a no-go or a suppressing pathway. The final structure that you might hear about a little bit, people call the hyperdirect pathway. It's actually a bit of a misnomer. This area here, STN, is the subthalamic nucleus. People often describe it as part of the basal ganglia. It's actually embryologically in the hypothalamus, which we talked about in the original macroanatomy lectures, is kind of like sitting on top of the whole brain and telling it what's really important. And so the subthalamic nucleus has this little, it's almost like a handbrake it can pull at any point in time. So you can imagine like this is the shusher going along, and the SDN is the sort of person that comes in and tells them, now's a really good time to shush. It's like this directed high frequency control over the handbrake. So this is just another sort of circuit diagram. You'll see a lot of these things. And the reason I like this one is that they kind of put it into this really nice model. So if you're interested, this is a really cool paper to read. Tim DeSteinen's group in eLife last year. And the idea is that if we take this little model that I've just shown you, right, where we've got a go pathway and a no-go pathway, or a facilitate and a suppress pathway, a really nice way to think about them is if you've got a little pulley which has weights on it. And if the weights are up, that means of the blue one, it means you can go. And if the weights are down, it means you stop. And so what your job then is with the basal ganglia is to pull this pulley around and to let certain things through and inhibit other things. So in a way, it's sort of like a filter for what the current processing mode is in the brain. And then you can imagine then the hyperdirect pathway, which is coming through here, it's going to link both of them up. Now we're not doing go or no go, everything's just turned off. So another really important aspect of these models is their relationship to a chemical called dopamine. So this is a neuromodulator. It's this sort of small little catecholinergic chemical. And the idea is that what dopamine is allowing the system to do is to balance the go pathway and the no-go pathway. So you won't see this in a lot of textbooks, but essentially when there's no dopamine in the basal ganglia, it turns out that the D2 pathway is like super overactive. It's firing like crazy. It's shutting everything down. And the no-go pathway is basically turned off. And when you wake up in the morning and you have your morning coffee and your dopamine levels start to equilibrate, what that has the effect of doing is pulling the system into this balanced state, such that now that any small movement, a little squirt of dopamine is going to pull the balance up so that you're now a little bit more go and a little bit less no-go, and vice versa. So it's about these neurochemicals are about bringing you to these steady states where small movements, the gradient of the change in a chemical is actually going to have an effect on the system's dynamics. All right, so this is another really great paper if you want to dig into basal ganglia stuff by Goldberg and colleagues in Trends in Neurosciences a couple of years ago. They were trying to grapple with this really funny thing that people have noticed in basal ganglia studies. And so this is, again, another example of the same kind of thing we've seen before, cortex activating striatum, which inhibits the globus pallidus and lets the functional circuit to occur. And you can imagine that if we were to represent the red, here this is going to be the globus pallidus, and then the thalamus here in blue as a sort of spike train. This is just a sort of an example that the globus pallidus will be firing, And then if I tell it to be quiet, then it'll open up this channel and allow the thalamus to fire. And then when it's done, it'll close the channel again. So this is this basic idea of kind of this inhibitory control over information processing. But there's a really fascinating thing, which is that there's these different modes that the system can be in. Sometimes it'll show these really weird rebounds where there's really no input to the system, but it just spikes anyway. There's other times when they seem to be really, really highly correlated, and you don't see an obvious gap. But in this paper, they basically argue that each of these different modes, even though they look different, are really just examples of this being driven in different frequencies. And so when you see the rebound, it's just that it's really, really low frequency activity, and every now and again you get a slip. There might just be a stochastic gap between some of these different inhibitions coming from the palatum that allow the thalamus to slip through. And when you're in that really high frequency mode where things are overlapping, if you actually unpack them in time, there's still an evidence of this little gap. So the basic idea holds even across all these different modes. All right, another important concept with the basal ganglia is that this same circuit that I've just shown you, this cortex driving the striatum to release the activity of the globus pallidus and also balance in the hyperdirect and indirect pathway, is spread out across the frontal cortex. The idea here is that here we're looking at a macaque brain, which is a non-human primate. And so the basic, you can see that the brain sort of looks a little different. It's a little bit less convoluted than the human brain we saw before, but the same basic architecture's there. You can see the temporal lobe here, the occipital lobe, the parietal lobe. Here's the sort of sensory motor strip, and here's the frontal cortex. And the idea is that across the frontal cortex, if we move from the motor strip forward to the orbitofrontal cortex, you can see these same circuits projecting down, a cortical projection down to different parts of the basal ganglia, which then through the pallidum, again with the same architecture conserved, disinhibit the thalamus and allow it to communicate back up in that same zone where the original projection was sent. So this is a sort of a very popular way of thinking about how the basal ganglia is organized at a system level. And the traditional way to interpret this was that these were kind of like what we would call parallel segregated loops. So you can imagine something like if the motor cortex decides that now is the time to wiggle your finger, then what it does is it says, hey, putamen, now's a good time to wiggle the finger. It'll release the thalamic channel, that'll come back up, and it'll let that system do its thing. In a second, I'll get to why that's maybe not the best analogy, because if you go and zoom into the anatomy, there's some things that are a little inconsistent with that model. But the basic idea, I think, holds up, that you've got this frontal gradient of control that can be mediated through this very particular architecture of inhibition and inhibition inhibiting inhibition in the basal ganglia architecture. And so, so one of the really interesting areas of research that's really unpacked what the basal ganglia is able to do has looked at the basal ganglia activity and the function of these circuits over the course of learning. So what they'll do is they'll get a little rat in and they'll ask the rat to learn button press gives you a food reward, and then over time the rat goes, oh, okay, I've got it, okay. If I do the little light, I push the button, I get the food, everything's great. It turns out that early in the course of learning, it's really, really important for that rat to have its caudate nucleus, but it doesn't really need the putamen. So the idea here is that something like the dorsolateral prefrontal cortex into the caudate is really crucial for it to learn this weird thing where the lights come in and it's had to decide what to do in that context. But the putamen's not really that important. This thing that's sort of releasing these particular motor plans. Maybe you could think about it like the animal doesn't really matter if they push with their right paw or their left paw, or they turn around and whack it with their tail. The important bit of information for them is to link the thing in the world with the particular outcome that they need. But then if they track these animals over time and they become better and better at the task, they have to do it with less and less deliberate behavior. What they find is that the motor cortex and the putamen become much, much more important, and all of a sudden the caudate isn't important anymore. So the idea here is that over the course of learning, you're shifting the control of this stimulus response or action outcome pairing from the dorsolateral prefrontal cortex and caudate further back down that little gradient I showed you to the motor cortex and the putamen, and allowing the putamen to sort of like take over what they would call the automatic process. So this is the basic idea, and there's lots of really beautiful evidence for this. one thing that I think a way to look at this that I find really appealing, it's certainly not the only way to think about it, but one thing that's changing, right, across this gradient is something like how controlled it is. And so this is maybe how we've operationalized it. Early on, you need to control your behavior, and over time it becomes a little bit more automatic. But there's another really important thing that's changing as a function of this learning, which also mimics the neurobiology. Early on, when you first see a stimulus and you don't know how to contextualize it, it's maybe you can imagine you've never played lacrosse before and someone gives you a lacrosse stick and just looking at it like, what on earth, there's like a net on it and part of it's got a little bulge to it and you don't quite know what to do with it. It's this really foreign thing. And you have to think about that lacrosse stick in the context of all the other things around you. But over time, as you start to play lacrosse more and more, now it becomes much more automatic. You know, if you push it this way, the weight will shift in this manner. And if I have a ball in it and I flick it around, then the ball will stay if I flick it at this frequency or not. In some sense, I've learned how to incorporate this tool into my world, in my body map. And so Hikosaka and colleagues argued in a paper, which I think is really beautiful, and if you're interested, you should have a read, that you can think about this as going from controlled to automatic, but also as going from world-based to ego-based. You're going from understanding the item that you've seen with respect to everything else around in the world versus the item with respect to you and the things that you can do with your body. And I think that these two things are not mutually exclusive. We could think about maybe controlled processes are what we need to use when we don't quite know the degrees of freedom of something. But I really think that's a nice way to think about it and can help you to kind of otherwise not get lost in all the circuit diagrams. All right. Another really important part of the gradient that I've showed you is that it's non-linear. There's directions baked within it. And this is by no means exhaustive, but a very beautiful paper, so a while ago now, almost 20 years ago, which is just terrifying to think about. Susan Haber and colleagues showed that if you track the connections from the globus pallidus down to the areas in the brainstem that are controlling the dopamine that comes right back up. I didn't show you this before, but when I showed you the output of the basal ganglia, there's a globus pallidus internus and also an area called the substantia nigra pars reticularis. This is kind of like a globus pallidus-like structure, and it's sitting right next to something called the substantia nigra pars compacta, and it's very different. It's this area that's packed full of dopamine cells. What these dopamine cells do is they take the chemicals from your diet, the amino acids, they apply certain translations to them, and they turn them into dopamine, which you then, your substantia nigra then projects up and basically douses your basal ganglia with dopamine, and then it's that level that you can control to open the channels or close them. It also, there's another part here, the ventral tegmental area you'll see, which projects to certain parts of the striatum, but also up into the frontal cortex. And it's really important in things like decision-making and motivation. But the thing that Suzanne Haber showed, and I think is really quite beautiful, is that if you look at the projections that come down from the more ventral portions of the striatum, they tend to promote the dopaminergic innovation of more, more caudal regions of the basal ganglia. So the idea is that anytime, and this is, certainly an interpretation rather than anything direct, but the idea being that anytime you become really motivated about something, you also want to then activate the systems that can help you make a plan about it. How can I get that thing? And then by the time you've built into a plan, now you want to know, how can I actually execute that plan? And so the idea is that there's this little spiral looping down that takes you from the goal you have to the plan you might want to have to the motor execution of that plan. And so it's almost like this this pull driving the system from these motivational states to action, which is, I think, a really nice idea as well, and maybe helps you think about it. All right, so I've showed you some pictures of the striatum. So this is just sort of another diagram to show. We've got a little late-defined pyramidal cell that I showed you in the cortex. And I remember I said that it sends off these little efferents, copies, and projections down into the subcortex. And it's going to hit these areas in the striatum. So here I've shown you the sort of little, the little telescoping Russian dolls kind of idea, where the striatum can inhibit the globus pallidus externus or internus. And then the globus pallidus internus will be the main output of the basal ganglia, which is going to come and then disinhibit the thalamus. So you also remember that I told you that the traditional model that people described was this idea of these parallel loops. So you can imagine that there might be a region that projects down that might then go and hit these really specific core thalamic cells and let the system resonate. Now the problem with that is that if you look in the neuroanatomy, that's not what's happening. These globus pallidus cells predominantly contact these matrix cells. The cells that I told you are much more diffuse, projecting up to the supragranular layers. So this idea of the basal ganglia unlocking a motor plan or unlocking an action are really not consistent with the neuroanatomy. What the neuroanatomy is telling us is that anytime the basal ganglia gets to do its thing, it's going to come up and it's going to turn up the temperature of that cortical area. It's going to increase the ability for the feedback to matter, for you to shift out of what you might have otherwise been your plan. And I think there's some really interesting ways to think about this that maybe explain why things like hyperdopaminergic states are associated with more variability rather than more consistency. It's also consistent with this notion, another beautiful paper by Paul Sisek, that shows that if you track basal ganglia, they actually do direct recordings from monkeys doing a set of cognitive tasks, that the basal ganglia is not telling you which target you're going to reach for, but it tells you how long you wait before you commit. So it's basically this little structure that's saying, hold off now, hold off now. It's too inconsistent. The wait is not in favor of option A or option B. Wait. And then when the evidence can become beneficial for option A versus option B, that's when you finally release this and you let the system go. And, you know, not shown here is obviously the connections that come down to the brainstem, which are much more specific, and I'll let the action plan out. So I think that's a really important point, and this is something that really, you know, hasn't been explored to date in the literature in any great detail. And then the last thing I want to say about the striatum is that there's a really interesting pathway that gets way too little attention. And so so when I showed you this little map here, we've got this idea of a loop where we've got a cortex coming down the basal ganglia and releasing some thalamic cells to come back up and project to the cortex. But what a large proportion of these little matrix cells do is actually project right back into the striatum and douse it with glutamate, right? This excitatory neurotransmitter. And so in some sense, there's this really interesting little loop that can occur where, you know, neuromodulating the thalamus might raise the temperature of these little cells here, which can then come in and cause this circuit to work either more effectively or less effectively, depending on which circuits it hits. And I think that's another area that's really important to think about. So this is just an example of that. You've got the putamen coming down to the globus pallidus internus, and then it hits this nucleus called the centromedian, which is at the matrix type, and it projects back into the putamen. And here's the paravesicular nucleus, both the frontal, the dorsolateral, and the rostral, doing that same kind of a circuit. And we don't really yet have a great handle on exactly what the importance of these different structures are for the function. All right, I'm just going to pause there for 2 seconds. Who's got questions about the basal ganglia? James.
So I think the idea of going from world to that would be stronger extension of our body.
Yeah, it's a really interesting question. Another thing that, so there's lots of animals that can use tools too, right? And one of the things that they have that's homologous to humans is a big cerebellum. And I'll talk about that in a sec. So maybe see how we go in the second half and see if that answers your question. But I definitely think, so the main areas in the frontal cortex, or in the cortex that have changed in humans, we can go back to the macaque, but there's this relative expansion of the lateral prefrontal cortex, what we call the granular prefrontal cortex, the lateral parietal, some of the areas of the temporal lobe, and then big areas of the cerebellum, the pons, and parts of the thalamus. But so if we just stuck on the cortex and kept the corticocentric view, as much as I disdain it, the idea would be that we've now got all this extra architecture around to do stuff with. We can draw in dots in interesting ways. And so there's a really cool idea that you actually might dig from Earl Miller's group, that what the prefrontal cortex is doing is playing a role like a reservoir computer, where it's just increasing the dimensionality of any signal that gets sent. And then you can use that dimensionality to then arbitrarily, you know, distinguish things that were otherwise really kind of similar to one another. You can be like, oh, actually, the branch that has this thickness is the one I should use to get the ants rather than that thickness, because that will snap. And so you've got this high dimensional kind of ability to deal with the world, which you can then relatively automatize. So in a way, I think the cortex is important, but it's only important because of all the other structures that it has with it. Eli?
Kind of alarming what you came to say. We're talking about primates and humans, and I agree that the development of the frontal cortex. I think I see what James is trying to get at. Like, is there evidence that, for example, basal ganglia, like developed to compensate. If you want to have this like a spiral down the hierarchy, where you start with the feedback loop has a little bit of abstraction, and abstraction comes back to reality. You move down, is there even for expansion, basically?
Yeah. Yeah, great question. It is definitely increased in size, just as well with the organizing principle in the cortex and cerebellum. But depending on who you ask, it's grown less than other structures. So what you have to do is you have to take into account brain volume, and you have to take into account the changes that have occurred across the whole neuroaxis. And you have to ask, relative to other structures change, have these changed more or less? I've seen some people argue that the globus pallidus externus has changed more in humans rather than non-human primates, suggesting that half of what we're trying to do is just make everything quiet so that we can selectively do the one thing and not others. But there's more evidence for lateral cerebellum, pons, lateral prefrontal cortex than the other structures. But it's still like an ongoing field. Last one, and then we'll keep moving.
Yeah, the kind of actually about going from this more, I guess, spatio-temporal driven, more ego-driven, and sort of saying that that's really automatic or becoming more automatic. I think that's what sort of fits with this idea, which is more kind of that kind of cognitive map type idea that, you know, when you're sort of first learning things, you're quite sort of relying on, you know, association with what's going on around you, but then when you get better at it, it becomes sort of more invariant to context, right? So you can be sort of more higher up sort of task structures or structures in the environment like that, so you can sort of deploy them anywhere.
Yeah.
So that, yeah, which is, I don't know if that's exactly automatic, it's sort of becoming more automatic, but it's becoming, I guess, more about learning the kind of like big meta level task structure.
Yep.
Invariant.
It's an excellent point, and it goes to show that there are many, many different ways to characterize the change that occurs from something you've just seen for the first time versus something that you now know quite well. And it could be something like the dimensionality of the signal, it could be how controlled or deliberate it was, it could be how external versus bodily mapped it was. All of these things need not be mutually exclusive, and they all are probably just different ways to describe the general phenomenon. Okay, I'll save other questions for the end if there are any. So the other structure that I want to talk a little bit about today is the cerebellum. And I like to call this the forgotten structure at the base of the brain, because this is a structure which has a literal boatload of neurons. In fact, the majority of neurons in the brain live in the cerebellum, which, I mean, if you were the cerebellum, you'd be kind of pissed off. When the anatomists describe it, it actually literally means little brain. It's like, come on, man. Like, you know, I'm doing all the work and I get no credit. So if we put these things side by side, so here I've got the cerebral cortex and the cerebellar cortex, and then I flatten them out, so that I've now basically like just sort of, you can imagine just pulling them all apart from their different dimensions. And so you can track and you know, look at different structures in here, but I think from memory, this is something like the occipital pole here. As you move towards, this is the insula, which is that little hidden area, and you might have like frontal over here, and I think temporal and parietal over there. Whereas the cerebellum, you know, ripped apart, it's much smaller in surface area, but it's very, very, very highly what's the word? It's sort of gyrophyte. So it's got a really, really sort of packed cellular volume. And so you can stack these things up against each other, and you'd say, well, the cortex has got, you know, 80% of the mass, so it's the fatty, but the cerebellar cortex has got 69 billion neurons. It's got 75% of the neurons in the brain. So they're really different kind of operating principles for how they're organized. And the one that I want to focus on today is the fact that the cerebral cortex is really defined by an interconnected architecture where a pyramidal cell in one column can easily influence many other cells just around it. Whereas in the cerebellar cortex, there's a really defined modular architecture. And in fact, if you go and look at cerebellar circuits under a microscope, you see this exact same architecture repeated again and again and again and again. It's actually really quite beautiful to see. And the basic architecture, again, don't get too much of a headache looking at these things because they're just sort of wiring diagrams. But the basic idea is that the cerebellum, every part of the cerebellar cortex has these little parallel fibers running through it that kind of look like telephone lines. And they're below them, they've got a set of different nuclei, which are either feeding information into them. These are the axons of these granular cells. And they've got these Purkinje cells, which are basically listening into those telephone wires and determining how to shape the output of the cell, of the cerebellum, which goes to this thing called the deep cerebellar nuclei. So the idea is that you've got information coming from the cortex that will go through the pons, This is going to be that efference copy I talked about before that comes from those layer 5 pyramidal cells, or via the red nucleus or via the spinal cord and the inferior olive into one of these two input channels. And then they're going to come in and differentially affect either the telephone wires or the CIA listening in, and then together they're going to shape the output of the cerebellar nuclei. So just showing you this in another form, here's these afferent inputs coming in from the pontine nuclei coming up in the granular cells into what we call the mossy fibers. They come up, those granular cells go up and have these big projections, these parallel fibers, the telephone wires, and then you've got these Purkinje cells, which are basically listening in and doing that shaping of the deep cerebellar output. Now, the reason I put this in is it shows you the big difference between these two input streams. The pontine nuclei coming up and are relatively subtle. They're coming in and they're just sort of delicately touching this neuron and that neuron in the granular cell layer. And the really important thing here is that the granule cells make up the bulk of that 75%. So in fact, they're these tiny little cells, but they're actually the majority of the cells in your brain. And there's a ton of them. There's a diversity. They've got different time scales, different sets of parallel fibers. And there's this huge diversity of these different cells. In contrast, this inferior olive input doesn't have this subtlety. It comes up and it grabs one of these Purkinje cells and it just wraps its activity around it. It almost looks a little bit like the glomerulus in the kidney if you've ever seen an electron microscope. In the kidney, you have the kidney sitting there with its little glomerulus, which is the little filter, and the blood vessel comes in and wraps around it, maximizes its surface area to volume ratio, so that any blood that comes through the kidney is going to make contact with that glomerulus and then get filtered so you can turn it into urine and extract the waste. Very similar principle here. This inferior olive neuron really, really, really wants to make that Purkinje cell fire. So this, as I said before, this microzone, this basic concept is repeated ad nauseum all the way across the cerebellum. So it's an incredibly modular architecture. Remember I said before that if we went from the sensory cortex and the cerebral cortex to the association cortex, one second, it changed abruptly from a granular to an agranular cortex. Here, no matter where you are in the cerebellum, you're going to see the same architecture. Chris?
It's like a very feed-forward system. Is there any recurrence?
Yes, there is, and it's incredibly important. I'll bring that up in a tick. But yeah, it is very feed-forward. And in fact, one of the things that people ascribe to the cerebellum as a function is a forward model. So it basically says is, you just showed me the context with this efference copy. I'm going to predict the sensory consequences of that action and try to fill in the blanks for you. In fact, I'll skip forward a slide. So where these come from in phylogeny is actually really fascinating. I don't have time to dig into it, but this paper is so cool. There's a guy in New Zealand who basically shows that the cerebellum, as we know it now, isn't actually where the first time that a cerebellum-like architecture developed. In fact, it first developed in teleost fish, which then went on to become sharks. And what the cerebellum was doing in those sharks was it was taking, instead of the input, those inferior olive inputs, remember I told you that come in and hug the Purkinje cell, which is controlling the output. In humans, these come via the inferior olive and get filtered through the cortex and various other structures. In a teleost fish and the shark, this comes directly from the sensory input. So it'll come from the skin on their body, or maybe come from the eyes. But the shark, because it really, really, really wants to catch the prey and it really wants to eat it and go off and become its little dominant self, really, really, really needs a sharp signal that comes in. But the problem is it's got all this other bodily noise going on. It's breathing, it's heart rate, everything's kind of like clouding this signal. And anyone that's ever tried to fall asleep when they're have had too much caffeine, you can like hear your, the pulse in your ear. That's exactly when this is starting to fail, right? So what happened in these sharks is that they took this sensory input. What they were able to do is take the efference copy from their body and cancel it out. They were able to act like an adaptive filter, so that the output was purely the sensory input and none of the actual bodily noise that came in. So this might be a kind of explanation for why sharks, can smell blood, 10 kilometers away or whatever in the water, because they're getting pure sensory information that's uncoupled from all of the noise in their system. So this is a beautiful paper, and if you're interested. So then the idea is that over evolutionary time, maybe there was a gene duplication event. Now we've got two copies. The idea of the Montgomery pictures is that one of those copies became shrunken down in mammals and became part of our auditory system. But in the second copy became elaborated and then became stuck into the the corticocerebellar architecture. And so then the idea is that you can then extrapolate this out, right? Instead of the sensory input coming straight in, and then canceling out the activity that you got from the cortex. Here instead what you're doing is saying, here's the cortical context. Tell me what the best outcome should be, not to cancel out all that input, but to guess ahead, almost like in language process, like an autocomplete. What should the next step in the path be? And this is where the recurrent connections really matter, because all these deep cerebellar nuclei, as they project out to the thalamus, are actually coming back and projecting back into the parallel fibers. So this is, really, really new stuff, but there's a group in the University of Texas that have shown that you can condition an animal to learn long sequences of events on single stimuli. So via these specific paths from the cerebellar nuclei back up to the parallel fibers. So the idea is that the cerebellum isn't just like an input-output structure, it's an input and elaborated output structure. All right, this is just a quick point. The way the cerebellum learns is really different to the cortex too. In the cortex, you typically get, we didn't cover this a lot, but you get this thing called long-term potentiation that typically occurs when two cells fire in the same little window, so long as the external milieu is in the right, there's the right kinds of neurochemicals around. And you get long-term potentiation, which is the two synapses coming in and forming a bond. The exact opposite thing happens in the cerebellum. You can think about this Purkinje cell, the CIA, listening into all the parallel fibers it can. It's spread its activity out and it's listening to as many as it can. And then what the cerebellum then does, instead of creating long-term potentiation when it receives some kind of a rewarding signal, it actually breaks the connections on a couple of those little paths, and then allows then that difference that makes a difference is the thing that then shapes the activity of the deep cerebellar nuclei. The reason that can make sense, I'm sort of a bit out of space, out of time here, is that sorry, I'm a bit out of order, is that the Purkinje cell ends up having an inhibitory effect on these deep cerebellar nuclei. So it's a little bit like the globus pallidus. It's sitting there tonically active, telling the deep cerebellar nuclei to be quiet. And then depending on the particular pattern that you learn that comes in through these mossy fibers up into the granular cell layer, depending on the particular connection that you silence, now all of a sudden, instead of all of them being inhibited, one of them gets active, or two or three. And so those two or three are then going to stand out against that background of inhibition and then be propagated through the circuit. So it's a different mechanism, but the idea is kind of similar to the basal ganglia. All right. So a really interesting point, I don't have anywhere near enough time to unpack these things, but hopefully I can just sort of point you towards interesting papers to read. One of the things that the cerebellum does really well, and it does it via this granular cell layer that I was telling you is like super rich, full of little different cells that have tiny little differences between them, different time scales, different projections. The idea is that these circuits are set up for pattern separation. And this is a really beautiful paper that shows that there's a direct homology between the organization of the cerebellum and the organization of the hippocampus. So the way that we haven't gone into a lot of detail on the hippocampus, but the idea is that as inputs from the sensory system come into the hippocampus then spreads out and takes that signal and sort of over-represents it. It's like taking a vector image of a really crappy JPEG. You're taking the highest resolution copy you can with the hope that somewhere in that mess, you can then distinguish it from some other really similar item. So they call this a sparse distributed representation. And the idea is that the perforant path and the Purkinje cell, the parallel fibers, are doing something very similar, where they're actually separating out the inputs that they receive and taking a high resolution copy of a low resolution signal. Now, why this is going to be important is that we know that in the hippocampus, take James McClellan's work or Randall O'Reilly, they argue that what the hippocampus is trying to do is basically like find these little tags that give you very specific episodic memory, such that if I go through this little path versus that little path, this will reinstantiate that memory versus that memory. But all in the episodic domain. So I saw this in this context. Now, the cerebellum, based on how it's functioning and where it can contact, might be doing a very similar thing, but for actions and plans. So it's more a procedural type memory rather than episodic type memory. So it's really fascinating that we've got these structures in our brain that are set up to kind of do very similar phenomena, but in very different domains. And I think that's definitely worth thinking about some more. So Chris, you asked about you asked the question out of order, but this is the idea that this is if you're interested, that's the Eli paper I was mentioning, Michael Mork's group. The idea is that these deep cerebellar nuclei, while also projecting upwards, come back and give internal feedback both to the granular cells as well as to the Golgi cells, and basically allow the system to sort of continually learn sequences. A really important point to mention, and this is mainly kind of a historical point, is that for a very long time, and many would argue still, people think of the cerebellum as a purely motor structure. So we talk about it as something that's helping me balance right now. If I want to, wave my hands in the air, instead of my hands moving all around, they help me smoothly move them through the air. Right? The idea is that as I move my arm in this direction, I send a little corollary discharge to my cerebellum. It says, oh, you were trying to move in that direction. Here's the little boost to make sure you do this in the right way and you don't move all over the place. Anyone that wants a real intuition for this, the cerebellum is exquisitely sensitive to alcohol and GABA. And so anytime you get too drunk, the cerebellum gets switched off and you basically hobble all around. Well, like me, one time I tried to ride a bike home after a night out and yes, don't do that. So this is the basic idea is that the motor cortex would send this projection down the cerebellum and get some feedback by the thalamus. But it actually turns out that in humans, non-human primates as well, there's also some other really fascinating examples like the dolphin and some the New Caledonian crow as well as some other parrots. The lateral cerebellum, which is CRUS1 and CRUS2, which projects to the medial dorsal thalamus and the granular prefrontal cortex, is actually expanded in these lineages. And so we know functionally that these areas are really important in things like planning and cognition, but here they have this cerebellar architecture underneath them that's supporting and augmenting their functioning. Some people, Randy Buckner has a really, really nice review paper in Neuron that basically argues that the cerebellum is what he calls a spandrel. I don't know if you guys are familiar with that term, but it's an idea in evolutionary science that certain things that can look really special can have come along for the ride by accident. And a spandrel is the little area between an arch and the sort of ornate ceiling in a church. It's like in a little corner, and they put all these fancy things on it because it's just sitting there. It doesn't have a function. It's not holding up the roof. It's not a doorway, but it's there because it had to be there because of all the other things. So some people argue that the cerebellum is a spandrel. It's not doing any work. I think that there's plenty of evidence against that, but I'm happy to chat about that with anyone. One bit of evidence, this is just a really nice paper by a collaborator of mine. They actually showed that as you learn to automatize cognitive rules, this is actually a meta-analytic paper, sorry, I gave the wrong reference, but you can find in Josh's work. They actually show that there's these regions of the cerebellum that are strongly related into these areas like the parietal lobe and the frontal cortex that we know are typically involved in cognitive tasks. All right, so to close the loop, I showed you before this little picture. We had the PT-type pyramidal cells projecting down to the striatum, and they hit these matrix cells, which then kind of boosted this variability. And here's this little picture of our cerebellum where we've got this efferent discharge coming down to the pontine nuclei. These guys have also preferentially expanded in humans, which is quite cool. They project into the granule cells, which then have these telephone wire parallel fibers, which are then listened into by the CIA, which shape the deep cerebellar nuclei, and they come back and hit the thalamus. And I probably either made this way too obvious or not done my job properly if you couldn't guess that they're actually projecting to the exact opposite thalamic cells as the basal ganglia. So they're actually projecting to the core cells, the ones that we said before when they receive the visual or the auditory or the somatosensory input, but projecting into the granular layers of the brain. And so what this suggests is that you've got this architecture here where an efferent copy that's sent out from the cortex can differentially decide whether or not to boost some variability, some feedback, some localized context for what it's doing, or through the cerebellar architecture, boost a really localized and precise resonance within a circuit. And I think this is actually a really important organizing principle that we don't think about enough. So I'm not going to have time to really unpack this properly, but one of the really important implications is that when we think about automaticity, right? Remember before I mentioned that as you learn a task, it goes from being something that is in the world towards, maybe more motive-based or maybe something that's more deliberate, more automatic. As I said, there's lots of evidence for this. And so we can maybe imagine tracking a goal to a plan to a motor action. It actually turns out that if you overtrain animals, and I'm not talking about within a few weeks or a few months, the typical kind of lifespan of an experiment in the sort of modern grant-based age. If you train an animal for a year to 2 years on a particular boring little task, you get the light and you push the button. You get the light and you push the button. This is the most bored experimental animal in the world. It turns out that you can lesion any part of the basal ganglia you want. You can actually lesion large parts of the motor cortex and nothing matters. What's really important is that the deep cerebellar nuclei are intact. So the idea here is that you're actually transferring the function over time as you become more and more automatic from this system that really needs all of this rich variability that the cortex and the basal ganglia imbue it with, to something that can handle something on its own with a more modular architecture. Now, I think this makes a lot of sense, right? If we think about the way the cortex is organized, it's very interconnected. It does this unsupervised learning. It can learn any kind of pattern you give it, especially if you augment it with a little bit of shaping with the basal ganglia. But what it's going to be really bad at is doing the mundane task again and again and again. Let's say you try to do the mundane task, and I gave you a little bit too much basal ganglia feedback. Now all of a sudden you do something slightly differently, and you drive your car off the road. What you want to do is you want to put that task into an architecture that can handle these little sequences where nothing's changing. So I think this makes a lot of sense, and it's definitely not the way people think about it in the literature. I can tell you quite frustrating. Anyway, so the idea in cartoon form is that maybe early in learning you've got a lot of cortex and basal ganglia, but the cerebellum's trying to learn what's going on, and then as you become better and better at the task, the basal ganglia is really important. Cerebellum is trying to learn that signal of how to control it, but then over time, really all you need is the motor cortex, maybe a quick little kick from the basal ganglia to set you off, and then the cerebellum can play out the skill or the particular behavior that you've seen without need for any deliberate processing. All right, so I think I'm firmly out of time, so I'll just skip over a couple slides. I was going to talk about the neuromodulatory system, but we have to say that for another time. All right, I was going to say ask me anything, but you don't have time, so suck it in, you can't. So, summary. Hopefully, this was a kind of bird's eye view, whirlwind tour to disambiguate some of the terms you've seen in the literature, to try to think about how you could put the cortex with the thalamus, with the cerebellum, with the basal ganglia, and think about how they might start to interact together. If you're someone that thinks about one particular nuclei, I encourage you strongly to think about its context in the system. No single cell in the nervous system does anything if you put it in a dish. You and I were talking about this yesterday, right? I could take your visual cortex out, put it on the table, it's not going to be processing any visual information. So it's the context that really matters for these things. And I think we're making a big error if we think about these things in isolation and forget about their context. The second is, as I've said at the very start of this, if you really want to get a handle on this stuff and remember exactly the little details, you really do have to take a project and try to learn the details. But I'm more than happy to answer questions. So if you go away and listen to the lectures again, or I didn't cover your favorite area, drop me an email. I'm happy to answer any questions. And with that, I'll say thank you. Cool. If people want to ask any questions, they definitely can, but don't feel like you need to hang. Questions? Go.
I know the cerebellum, which another kind of great way I've heard it referred to is a pillar of the cortex. That was what someone described to me when I told them I was organising a conference on the cerebellum.
Oh, nice.
Yeah. So I know one way people kind of attack, or a lot of people attack the cerebellum is kind of observing this really recurrent modular architecture. So it looks the same all the way across. So the whole thing must be doing the same thing. So what's the kind of silver bullet of the cerebellum? Do you think that's the kind of right way to think about it? So all these things we're talking about with the cerebellum, is that something that we kind of consider the cerebellum is doing as a whole? As it does one thing, of course in different modalities, but it kind of has one job and kind of operates in one way.
Yeah, so there's definitely some pushback against this idea. There was a recent editorial by Rich Ivory's group in Neuron that kind of argued that maybe the non-linearities that exist within the circuitry might have some impact that could kind of make the circuit less modular than we think it is. So for one example is that the pontine nuclei, which are feeding in the activity of the granule cells, have really weird connections between them. So it's not just like a blind relay where it sort of receives the efference copy and sort of throws it onto the next thing. It's also inhibiting friends and doing long-term potentiation and depression. So there could be a kind of non-linear filter there that is different for cognitive versus motor versus emotional tasks, right? But when it boils down to it, the cerebellum is an incredibly wise individual that can't really do much other than kind of poke you and say, you probably should do this. So I was actually using this analogy. I think the analogy of Yoda with Luke is really good, right? You've got this like wise old man or whatever the hell Yoda is telling Luke how to be a better Jedi. And Luke kind of ignores him for a while. And all that Yoda can never do is like tell him you ought to do it this way. And Luke is free to ignore that. He can go off and do his own thing. He's better off when he does listen to Yoda, and he actually benefits from having Yoda's input. But at the end of the day, Yoda can't do it for him. He can't, get in the bloody X-Wing or whatever. So I think that the cerebellum really has only one way to affect things, which is by boosting the core thalamic nuclei and boosting parts of the red nucleus. That's its output, right? And so even if it is doing different computations, those are probably different contexts in which it's working in. I personally think it's quite appealing to think about cognitive automaticity within this lens. Think about an expert chess player. When they're playing the game, they're thinking about this really high-dimensional state because everything else is automatic. They know the rules. They know the context. They know what the other players thinking in this context. That all falls away. Because maybe the cerebellum is boosting and going, you know what, we've seen this 100 times before. A little heuristic, intuition. But then on top of that, they play the little deliberate path that selects that move and not that move. So I actually think that this helps you to reframe what cognition might actually be and why humans are a little different than other animals in terms of our cognitive repertoire. Because we can automatize way more things and everything isn't just stuck up in our mind all the time. It's just playing out for us automatically.
Like, compelling idea when you think about resource management as well. You have the circuitry that's great for algorithm learning. Once you actually figured out, this works pretty well, the solution, farm up some other reason, you free that circuit up again and then explore some new axis of improvement. Sports is pretty good all around, second order.
How to curve the ball or
things like where players might be and different combinations. Even chess players talk about movements and talk about intuition engine.
Yeah.
Higher order effects, second order. Momentum is a term that they use to describe sport.
So if you were going to describe this in Bayesian terms, you might say it's a prior, right? And this isn't typically the way we think about Bayesian computation happening purely in the cortex, with a top-down thing saying, I guess this will happen, and a bottom-up saying, did it happen or not? The cerebellum is like this little friend that goes, oh, it's probably going to be that thing, buddy, which is going to be something like a prior, but it's going to be a different kind of a prior than you get from the cortex. And this is one of the really interesting questions is, how do you map things like active inference onto the neurobiological the way that it actually is, rather than the way we kind of wish it would be if we kind of guessed it first. Anyway, that's what we were talking about. Any other questions? Any favorite regions I didn't talk about that people kind of want to hear about or no? Cool. All right. Thank you.